Where did Christian go? Christian! So, we just went to the bigger park in town, and they literally have it taped off. All the equipment they have taped off. And thank goodness we know lots and lots of creeks for us to go to. If these people, they think we're going to sit inside all the time while this is going on, they are out of their minds. People have lost their minds everywhere. We just went 2 different public places and people are freaking out. We went to the health unit to pick up some paperwork, and the nurse there was like standing way in the corner. They won't even hand paperwork through the window to people. This is oh my God, like I keep thinking to myself, let's just imagine, what if this theory, this germ theory, is completely wrong? Completely wrong. Or, what if there's just something we're missing? We're missing the boat here on some aspect of this. Maybe they're contagious, but just to, obviously the terrain being as bacteria and these microbes and whatnot can change with the environment. The terrain means a lot, obviously. Yet we've got a whole world of people, supposedly educated people, who are just phenomenally hoodwinked. It's like black magic. That's what I see. I see a world just under a spell of this magic. It is incredible. And even, let's just throw all that out and let's say it's real. COVID-19 is dangerous. It can be deadly. It can strike anywhere. It doesn't matter the terrain. Let's just say the threat is real. Regardless, even with this contagious virus going around, the numbers are not adding up regardless. So the hysteria is just uncalled for. It's uncalled for. So I noticed that Michelle Lowe made a video talking about Frank Tufano's conspiracies. Now he's saying that viruses aren't contagious or aren't real or whatever. And of course, when he speaks on these things, he acts very certain, as if he's been studying this for, years, which he hasn't. This is why I cannot speak certainly on this. I can look at what we do and don't know, and I can speak on that. But she was saying that how it's just so disrespectful. It's so disrespectful, and this is the sort of spell I see people under. This is what I'm talking about. How is this disrespectful to look for things yourself, to question things and try to understand them yourself, instead of just having faith in the system, faith in other people's study, other people's beliefs, just because there's a consensus. It made me think, okay, so imagine, the 18th century, let's say there are cholera outbreaks everywhere, people are pooping in the street. Do you think those people living during that time, do you think they knew they lived in the quote-unquote dark ages? I would bet you they didn't know that. I would bet you, they just had no idea about what they didn't know. They had their own theories and their own modern ways of thinking. And how did we get to this conclusion? these things are mostly based off of theories and assumptions, and then you just put your faith in that and go from there. So, what was it, Louis Pasteur? He was basically poisoning animals with, toxins that anaerobic bacteria made, but at the time, he was using these filters and he was filtering through. He realized there was something else other than the bacteria that weren't being filtered through. There was something else that were causing a sickness. And so they decided from there that every one of these little viruses, every one of these little bugs were attributable for this certain type of disease, whatever type of disease they caused. And at some point this was just decided upon that this is what it was. They were contagious. Every virus had ailments to go with it, and these were just random happenings, and you would get these things that were contagious. And that was that. So everything is based off of the theory, and you have to have complete and utter faith in this initial theory and go from there. And then everything from there seems to be mostly man-made, is what I'm gathering. We do see that certain cells, aren't susceptible. They have to have specific receptors and things like that to take in a virus. So we see that we can't see all the viruses, first of all. We can't study them under a microscope because we can't see them. We can see the larger ones, and from those we can grow. But everything, that's the thing, even if we can grow viruses on a petri dish, in Virology 101 they will teach you that not all cells will take in a virus. They have to have receptors, they have to have the machinery to duplicate them, and they say it's random for the most part because they don't know. I can guarantee you it's not random, but we don't know. That's the thing, we don't know. All we can do is put a petri dish down and watch the growth. You can have all of these man-made terminologies and ways to perceive how this growth is happening, and from that I think you can conclude that there's some sort of contagious aspect to this, that they can get in some of the cells. I think that's safe to say. But we're not looking at every step to this process. We're not isolating and seeing. I hope I'm explaining this well enough. Okay, so from there we build up these man-made systems. That's what I'm saying. We take, based off of a picture, based off of a particle, we build up the genomes to these quote-unquote viruses, and then we have a system we can look at when we need to go back in and test people. We can pair it up to something and say, okay, it looks like.
Demonstrate that scientifically sound evidence for the existence of the viruses that supposedly cause the avian flu, cervical cancer, SARS, BSE, hepatitis, AIDS or polio has never been provided. Quote, as with HIV, isolating one of these viruses in a purified form with its complete genetic material and virus shell directly from it and then imaging it with an electron microscope has never taken place. So let's look at some of these images of viruses that circulate on the internet and in medical journals. Okay, these are just a few examples. You can Google this yourself. Just Google hepatitis virus and so forth. And that is what we find. Well, what are we seeing here? Well, most of these images look more than artwork than images of a suggested virus. Also, electron microscope images usually appear as grayish or black and white, grayish, brownish, olivish, and so forth. Also, there are different appearances of these viruses, as we can see. So this one is from a German paper, looks specifically scary, doesn't it, right? So these are pictures or images of the so-called swine flu virus. Okay, we see they all look a little bit different, okay? So whether colored or not, The question we need to ask ourselves is, are these actually pictures of viruses? Okay? Answer, Lanka. The photos of the alleged viruses are not showing an isolated virus, but cells or cell particles. Virus Mania authors. The alleged viruses are in reality micro.
I'm also going to leave in a couple clips here at the end on how, they determine how these cultures are growing in a lab setting, how the testing's being done even with like COVID-19, and how they use this database and things like that of this man-made system that we've created. Not saying it's incorrect. I'm just saying, like, it's not a bad idea to go back and ask these questions, especially when the terrain theory really, seems to be, with the new evidence we're finding on bacteria, and the more we learn through the years and decades, we see that this terrain theory holds a lot of weight. It really does seem to ring true in a lot of senses. So, and like I've said before, I think both could be, have bits and pieces that are correct. That makes sense to me. But we're just, this modern medical construct is so arrogant, thinking, and people, everyone else, who just leave it all up to these brilliant scientists to figure it out. And these brilliant scientists, they don't even know. That's what people don't get. They are being taught the same virology courses that you can look on YouTube at. This is what they are being taught. They have certain techniques and ways of looking at these things without ever looking at the virus. And again, this is all based on the assumption that they are randomly, contagious and causing disease, when this doesn't seem to be the case. Once we know all we do now today about bacteria, about microbes, how they change depending on the terrain, depending on the environment. And this can be studied. This is understood now. This was not understood back then when the germ theory won out. Scientists don't even agree on where they come from. They agree that they come from somewhere external, that they're not made within our bodies, even though we can find them now in the genomes of every animal, every plant, every human. But they don't know when they came to be or how or anything like that. They don't know how old they are. They know that they seem to be as old as us or older, So that's kind of up in the air still. But yeah, the idea is that they are external and they infect us. And this infection theory, oh my goodness, this infection theory, if this is incorrect, oh Lord have mercy, help us all.
Viruses in. First, let's answer a couple of questions. First of all, how do you know if the cells are infected? I give you a nice monolayer, you put virus on. How do you know that they're infected? And there are a couple of ways we can do that. One is by simply looking at them in a light microscope and looking for what we call cytopathic effect, CPE. And that is changes in the cells that are visible through a light microscope. And so here we have pictures of HeLa cells in culture that are infected with poliovirus. On the upper left is the monolayer before it's infected. Lovely monolayer. All the cells are touching each other from one side of the plate to the other. And then we infect it. We take off the medium. We add a small volume with poliovirus in it. We let the virus attach. And then we add fresh medium. And then we incubate. And on the top right, something like 4 hours post-infection, 8 hours, and 12 hours. And I think you can see things are happening here. Let's go through what they did, because I've explained to you every step of this process. So they had a sample of bronchoalveolar lavage from a patient who had pneumonia. So they put a tube into your lung all the way down to the bronchi. They put in a little PBS, and then they pull it back out again. And then they say they did a combination of Sanger, Illumina, and nanopore sequencing. Sanger is the old style sequencing. The others are high throughput. So they take this sample and they extract the nucleic acids. Now, if they're looking for an RNA virus, they first have to use PCR to convert it to DNA, because these high throughput methods only work with DNA. They got the whole genome from this sequence, 15,000 bases, and they did phylogenetic analysis. Okay? So from a wash from a patient, the entire genome, phylogenetic analysis, that's shown here. This is done by the EcoHealth Alliance. And here's the phylogenetic tree of many coronaviruses, and here is the Wuhan isolate from December. So what they've done here you can see the scale. There's no numbers here to indicate the quality, but you can always get this information from the raw data. The tree is based on partial gene sequence, 400 base pairs of the RNA polymerase. The RNA polymerase is the only protein conserved among RNA viruses, so it makes sense to just look at that. There's a huge database of virus sequences that you can access and do this with. And look, here it is. It's right next to bat coronaviruses and the SARS coronavirus from 2003. So boom, that's immediately put in there without knowing anything about the virus. The sequence immediately puts it on this tree of coronaviruses. You know exactly what it is, very closely related to a bat coronavirus. So anyway, that's how they figured out that it was this new virus. Now there are tons and tons of new sequences arising, and as things emerge, I will tell you more about it.
These advancements in medicine are amazing, and one day we will have a microscope that can see as tiny as they come. You know, we can see all of these little viruses or whatever the hell they are. But yeah, let's go ahead and treat it as if it's real, because I mean we don't know. They can be harmful, maybe. I mean, just like these toxins that bacteria make when they become anaerobic, when they're outside of the body and they cannot get oxygen anymore. Now, sometimes I'd say this can happen in the body as well. Nutrient deficiencies and all sorts of things. I can see this happening, these bacteria not being able to survive, and then they become anaerobic, and just like on rotting food, rotting flesh, they can make you sick if you eat something with some of these pathogens on it. You can get food poisoning. So this is what Louis Pasteur did to all of his little experiments. These animals, he was poisoning them, and he was calling it rabies. He was calling it different viruses because I don't know, maybe he believed that's what it was. And, you know, if we could do studies on this sort of thing, of course, that would be unethical. Just the same way they say it's unethical to study the vaccinated versus the unvaccinated. You can't get people, purposefully get them sick and study. We could do this with animals, and studies that have been done with this, it didn't work. So, I mean, I don't know. It just seems like it's true to everyone. Even with myself, it seems like kids bring home sicknesses from school. But I can recall times when, for instance, my middle son, when he was vaccine damaged and we were at home for weeks on end, it was like his immune system was creating these things. He got hand, foot, and mouth and a couple of other illnesses. Of course, I will keep looking, and I will keep letting people know what it is I'm finding, regardless of how quote-unquote disrespectful it is, because we're supposed to have faith in the system. No thanks.